This module pertains to DNA methylation. All of the cells within our individual bodies have the same DNA. However, there is huge variation in the structure and function of the cells in our body. That is, we have hair, rods and cones in our eyes, hepatocytes in our liver, glomeruli in your kidney, etc. All are very different despite having the same DNA sequence. These differences between cells that have identical DNA sequences are passed on from one cell to its progeny. That is, liver cells continue to proliferate liver cells, kidney cells continue to proliferate kidney cells, etc. This phenomenon is known as epigenetics. In other words, epigenetics can be defined as changes in gene expression patterns that are mediated through mechanisms other than changes in the DNA sequence. Epigenetic inheritance is the acquisition of heritable change in gene expression that occurs without a change in DNA sequence. Epigenetic alterations allow genomic adaptation to environmental changes and insults. That is, epigenetic changes are modifications to DNA that regulate whether genes are turned on or off. These modifications are attached to DNA and do not change the sequence of the DNA building blocks. Examples of epigenetic modifications include histone modifications, non-coding RNA actions, and DNA methylation. In this module, we will discuss DNA methylation. Before discussing DNA methylation, it is important to return to the DNA sequence. A CpG site is a region of the DNA where a cytosine nucleotide is followed by a guanine nucleotide in the linear sequence of bases along its 5' to 3' direction, as seen in this figure. CpG islands are regions with a high frequency of CpG sites. Initially, a CpG island was defined to be a region of at least 200 base pairs in length, having a GC percentage greater than 50% and an observed to expected CpG ratio greater than 60%. Later, this definition was updated to DNA regions greater than 500 base pairs with a GC content greater than 55% and an observed to expected CpG ratio of at least 65%. In humans, about 70% of promoters located near the transcription start site of a gene contain a CpG island. Because of this, the presence of a CpG island is used to help in the prediction and annotation of genes. 5-methylcytosine, which is a methylated form of the DNA base cytosine, or C, is the only methylated base in human DNA. Its methylation occurs within the context of CpG sites. DNA methylation occurs when a methyl group attaches to a 5' cytosine and a CpG dinucleotide. The presence of multiple methylated CpG sites and CpG islands of promoters causes stable silencing of genes. That is, when CpG sites lie within a promoter element, their methylation generally corresponds to transcriptional silencing, which is assumed to have an effect equivalent to inactivating mutations in tumor suppressor genes. That is, hypermethylation of CpG islands in gene promoter regions is thought to affect gene transcription and is often called epigenetic silencing. In fact, CpG island hypermethylation has been described in almost every tumor type. The precise mechanism by which hypermethylation represses transcription is not clear, although empirical observations have led to the following two hypotheses: that methylation itself blocks the binding of transcription factors, and/or methylation modifies the chromatin structure, which in turn blocks access by transcription factors. Alternatively, loss of normal methylation in the promoter region can lead to a gene being expressed, which is ordinarily not expressed by that cell type, which is consistent with activation of oncogenes. Therefore, methylation of gene promoter regions is thought to be an early carcinogenic event. DNA methylation is a normal process that is tissue-specific. DNA methyltransferases maintain the methylation pattern during cell replication. In mammals, genomic methylation patterns are established during embryogenesis, through the interplay of at least 3 DNA methyltransferases. The latter two may act as de novo methyltransferases to establish methylation patterns during embryogenesis, while DNMT1 is thought to maintain these methylation patterns during DNA replication. Very little is known about how epigenetic alterations are induced, but 2 mechanisms have been identified for removal of methyl groups from CpG dinucleotides. One is DNA oxidation that results in chemical loss of methylation, and the other is enzymatic demethylation by the recently discovered TET proteins. Regardless, CpG methylation may be a molecular marker that may be independent of its functional implications. In general, methylation of CpGs in the promoter region of genes silences their expression, while demethylation increases gene expression. Because hypermethylation of promoters result in loss of gene function, cancer researchers are therefore interested in identifying genes that are also silenced by methylation in cancer samples, but not in corresponding normal samples. Moreover, the loss of methylation in cancers could induce unwanted expression of normally repressed transposons. Historically, around the years 2000, restriction landmark genomic scanning, or RLGS, was the high-dimensional assay used for identifying methylated sites and was based on an approach that selectively cleaves DNA by restriction endonuclease, which it specifically cleaves CpG islands. As mentioned, CpG islands surround the transcription start regions of a high percentage of genes in the human genome and are typically unmethylated. The cleavage products are reduced through further digestion by restriction endonuclease and separated by electrophoresis. Therefore, the resulting data are images of the gels. When comparing images of gels of paired normal and tumor tissues, intense spots represent co-migration of distinct fragments, and loss of signal is representative of hypermethylation. To identify the sites, the fragments have to be cut from the gel and cloned. For a patient with acute myeloid leukemia, we have enlargements of regions from their RLGS profiles at diagnosis, remission, and relapse. These spots were selected to demonstrate the different patterns of RLGS spot changes. For example, in one pattern, a spot is present only in the remission sample. In another pattern, a spot is missing only at relapse. This process of scoring and cloning is tedious and time-consuming. Subsequently, Illumina developed an array-based assay to assess methylation. Initially, this was the Golden Gate Methylation Bead Array Cancer Panel 1, which interrogates 1,505 CpG sites and 807 unique genes that had been previously identified to be associated with cancer. The cancer panel was on a 96-well plate, so 96 samples could be assayed at one time. We call the probes bead types due to the design used. Later, Illumina developed the Infinium Human Methylation 27 assay, which interrogated 27,578 CpG sites and 14,495 unique genes. Afterward, the Infinium Human Methylation 450K array was developed, and it was later upgraded to the EPIC array, which interrogates over 850,000 CpG sites. It is now in its second phase, which interrogates over 900,000 CpG sites. Regardless of the Illumina platform used, the technology for assessing DNA methylation proceeds by bisulfite converting the DNA prior to performing the hybridization assay. What is depicted here is a double-stranded DNA molecule with some CG dinucleotides methylated. After bisulfite conversion, methylated cytosines remain unchanged, while unmethylated cytosines are converted to uracil. After bisulfite conversion, the sample is whole genome amplified and enzymatically fragmented so that DNA is hybridized to the arrays. Remember, uracil replaces T in RNA, so the hybridization behavior of uracil is similar to that of thymine, or T. Therefore, you can detect the methylation status of a particular cytosine after a bisulfite treatment of DNA by using a genotyping assay for a CT polymorphism. Therefore, assays for methylation are adapted from high-throughput single nucleotide polymorphism genotyping systems, which are used in genome-wide association studies, or GWAS. On the left is an ideogram depicting how probes are designed to detect the methylation status of a particular cytosine after bisulfite treatment of DNA by using a genotyping assay for a CT polymorphism. Specifically, for a design probe that ends with an A, single-stranded sample will bind if a uracil is present because of the Watson-Crick base pairing rule. Recall, A binds to T. For a design probe that ends with a G, single-stranded sample will bind if a cytosine is present, again because of Watson-Crick base pairing rules, because C binds to G. Here, you can see a cytosine binding to the design probe that ends with a G, which indicates methylation at that locus, because cytosine remained unchanged after bisulfite treatment. Therefore, we minimally need 2 probes to detect methylation status of a given CpG locus. On the right, notice that if the sample intensity is primarily observed for the probe designed to end in an A, the sample is unmethylated at that CpG locus, because after bisulfite treatment, the unmethylated cytosines are converted to uracil, and so binding occurred. If the signal intensity is primarily observed for the probe designed to end in a G, the sample is methylated at that CpG locus, because after sulfite treatment, the methylated cytosines remain and bind to guanine. If the signal intensity is mixed, there is hemimethylation. That could mean that one allele is methylated and the other allele is unmethylated. For that reason, the initial Cancer Panel 1 and Infinium Human Methylation 27 arrays were designed such that for each CpG site, 2 pairs of probes were used, an allele-specific and a locus-specific oligo designed to match the methylated target sequence, and an allele-specific and locus-specific oligo designed to match the unmethylated target sequence. Both probes for the same CpG locus will incorporate the same type of labeled nucleotide determined by the base preceding the interrogated C in the CpG locus, and therefore will be detected by the same color channel. This simply means that probes were designed to interrogate the methylation status, that is, whether the sample is methylated or unmethylated at a given locus. Again, methylation protects cytosine from conversion, as you see on the right, whereas unmethylated cytosine is converted to uracil, as seen on the left. Illumina then decided to expand the platform to include more CpG sites. So with the 450K and EPIC assays, there are 2 ways methylation is assessed: using this original design and also including only 2 probes for each CpG locus, one for the methylated and the other for the unmethylated state. The 2 types of probes used to interrogate CpG loci on the 450K and EPIC platforms are called Type 1 and Type 2 probes. Because of unmethylated and methylated probe designs, for each sample, there are 2 resulting files that store the probe intensities. These are IDAT files, where the pair of IDAT files have names ending in red or green. Therefore, the assay is somewhat like custom spotted arrays, where for each locus, there is a methylated output and an unmethylated output. As with gene expression microarray data, there are several different normalization methods that can be applied to Illumina's methylation assays. This includes the method implemented in Illumina's Genome Studio software, which uses a reference array. Quantile normalization, which performs quantile normalization stratified by methylated and unmethylated intensities, where the distribution of type 1 and type 2 signals is forced to be the same by the first quantile normalizing and type 2 probes across samples and then interpolating a reference distribution to which the type 1 probes are normalized. Subset quantile within array normalization, or SWAN, which first creates an average quantile distribution, Using a subset of type 1 and 2 probes, then intensities on the remaining probes are adjusted for each probe type using linear interpolation between the subset probes. NOOB, which stands for Normal Exponential Out of Band, is a background correction method with dye bias normalization. Functional normalization is a between-array normalization method that removes unwanted variation by regressing out variability explained by the control probes present on the array, where the first K principal components of the internal control probes matrix play the role of the covariates adjusting for technical variation. Some of these methods were developed to handle the two bead types in the newer assays, that is, for the 450K and EPIC assays. Once the raw data have been normalized, we then extract methylation summaries. For each array I and B type J, the background corrected intensities per probe were used to estimate proportion methylated using this equation. That is, the quantitative amount of methylation at each CpG loci is reported as beta values. Beta values range from 0 to 1 and represent the percentage or proportion of DNA methylation present at a given site. In order to promote normality, the beta values are commonly transformed using the logit transformation. Because the logit of 0 and 1 are negative infinity and non-existent values, a beta value of 0 is commonly imputed to be some very small number, and a beta value of 1 is commonly imputed to be some very large number prior to applying the logit transformation. Before statistical analysis, filtering methods are commonly applied. This often includes removing the probes for which the detection p-value indicates the signal is no different from background, removing the probes that have been identified to include a single nucleotide polymorphism in their sequence, removing the probes documented in the literature to be unreliable, or removing probes that have small differences in the beta values across all samples. The minfi Bioconductor package can be used to read in raw data from Illumina's platform and perform preprocessing methods, including normalization and obtaining methylation summaries. Recall that phenotypic data provide information about the samples. For Illumina assays, there is always a sample sheet description file that should be used to read in the data to ensure the phenotypic data and the methylation data are properly aligned. Therefore, the minfi package includes functions for reading in phenotypic data and methylation data. First, read in the phenotypic data frame and look at the first several rows. The sample sheet stores the links to the methylation arrays, which includes two columns, Centrix ID and Centrix position. Taken together, these fields allow you to associate the appropriate hybridization assay to its phenotypic data stored in the sample sheet. The minfi package looks for a column in the sample sheet called base name, which is Centrix ID and Centrix position pasted together and separated by an underscore. That field was called well in our sample sheet, so we added base name to our phenotypic sample sheet as well. The read.metharray.exp function allows you to read the IDAT files where targets indicates the phenotypic data. Again, this function assumes that targets includes a field called base name that indicates the base name of each pair of IDAT files, where the pair of files have names ending in _red or _green. It may be helpful to examine the density plot of beta values for each sample. The density plot function produces density plots of the methylation beta values for all samples, typically colored by sample group. We typically should see that most CpG sites are either unmethylated or methylated. In this case, all samples look good. While the density plots are useful for identifying deviant samples, if there is a problematic sample, it is not easy to identify which sample has the problem. If there is a concern about outlier samples, a useful follow-up plot is the bean plot that shows each sample in its own section. Here is a bean plot of the beta values for each sample in our example dataset. The QC report function produces both density plot and the bean plot, as well as control strip plots, which plots individual control probe sets. The output is a PDF file written to the working directory. The report can be examined to determine whether any samples were of poor quality. Pre-processing or normalization takes as input the RG channel set and returns a methyl set. A number of pre-processing options are available. Each set of methods are implemented as a function with the name starting with pre-process, followed by the name of the method. Each method may have a number of tuning parameters. Raw preprocessing means simply converting the red and the green channel intensities into methylated and unmethylated signals. Because SWAN takes a random sample of type 1 and type 2 probes when forming the normalization relation, it is a good idea to set the random seed prior to using SWAN normalization so that you will be able to reproduce your results. Although all of these methods result in very high correlations among technical replicates, a recent paper that compared various normalization procedures for the EPIC array demonstrated that pre-process NOOB had the highest correlations among technical replicates and lower standard deviations. Another competitive normalization method is Sesame, which is implemented in a different bioconductor package and will not be covered in this module. Typically, some type of filtering is applied prior to conducting statistical analysis. A common filtering step is to exclude probes whereby signal is not above background levels. The detection P function identifies failed positions defined as both the methylated and unmethylated channels reporting background signal levels. A small p-value indicates a good probe. Probes with non-significant p-values, typically greater than 0.01, should not be trusted. Notice that the NOOB and detect P objects are aligned. This allows us to filter NOOB using results from detect P. We can construct an indicator to identify probes that are absent, that is, not different from background, across all samples, and then filter NOOB to remove probes absent in most or all samples. Here we keep probes for which absent is equal to 0, meaning that all probes are above background across all samples. For the EPIC array, previous researchers identified that some probes that were designed to interrogate selected CPG sites have poor performance in that they do not interrogate their intended target. The names of these probes can be downloaded and removed from the data prior to statistical analysis. The example data set here is from a 450K array rather than from EPIC. In the Active Learning Lab, you will gain hands-on experience filtering these off-target probes from an EPIC dataset. The normalized object is a methyl set. Once a methyl set has been generated, we have various ways of getting access to the methylation data. The most basic functions are getMeth and getUnmeth, which return unlogged methylation signal intensities. The function getBeta extracts the beta values, which are values between 0 and 1, with 1 being interpreted as very high methylation and 0 being interpreted as very low methylation. If type equals Illumina, which is not the default, these are computed using Illumina's formula, which adds 100 to the denominator. If you examine the methylated and unmethylated signals for the first sample and the first CPG site on the previous slide, we see how getBeta calculates the beta value returned. The default does not add 100 to the denominator. The function getM is equivalent to extracting the logit transform beta values, where the beta values do not include plus 100 in the denominator, and the logarithm applied is log base 2. These two expressions are therefore equivalent. After normalizing, obtaining methylation summaries, and filtering the data, statistical analyses are performed. The methods we discussed for supervised learning, including identifying differentially expressed genes using Lima, can be applied, although we use the terminology for these data to identify differentially methylated CPG sites. Also, prediction methods such as penalized models including lasso, elastic net, and ridge, k-nearest neighbors, classification and regression trees, and random forests can be applied, as well as unsupervised learning or clustering methods and dimension reduction methods. minfi does have its own function for identifying differentially methylated CpG sites, but you can also use the Illumina package. Here is a demonstration of minfi's dump finder function. The dump finder function takes an object, a phenotypic variable. Type is a parameter that indicates whether the phenotype is categorical or continuous. The Q cutoff indicates the FDR, calculated using the Q-value method, greater than the specified threshold will not be returned. And shrink var is a logical parameter indicating whether or not shrinkage should be used for the variance. In this analysis, 35 CpG sites were significant using an FDR less than 0.05. The get annotation function can then be used to look up information associated with these differentially methylated positions. Some topics are considered to be more advanced and thus are not covered in this course. However, mentioning these topics may be useful for those of you who plan on analyzing high-throughput genomic data in your research program. First, when studying peripheral blood samples, remember that DNA methylation is a normal process that is tissue-specific. Peripheral blood samples are composed of many different cell types, so the estimate cell counts function can be used to estimate the relative proportion of pure cell types within each sample. That is, given peripheral blood samples, the estimate cell counts function will return the proportions of lymphocytes, monocytes, B cells, and neutrophils. These values, or their transformations, can then be used to adjust differential methylation analyses. Also, there are now many different ways to use methylation data to approximate biological age. Several researchers have found interesting association between inferred biological age, which are more interesting in comparison to chronological age. Regardless of what high-throughput genomic assay you are analyzing, it may be useful to check for whether batch effects are present and adjust for any batch effects found. A commonly applied batch correction method is combat. Here are some related references.